Thank you, Cindy. The devil's advocate is gone, and now the patient's advocate is here. Okay, so my talk's called Morgellons Is It Infection? And again, this is my disclosure slide, and I do not have conflicts of interests. And by now we've heard this same definition a lot, but just in brief, it's a dermatologic condition associated with Lyme disease. It's characterized by those slowly healing, spontaneously appearing ulcerative filamentous lesions. And that's a real beauty there. You can see some of those filaments coming out of the skin and, growing upward and protruding from the edges of that lesion. And again, the key diagnostic criterion is the presence of the colored filaments, mostly white, blue, or red, that protrude from or are embedded in skin. Now we're going to go over the history again, but this time we're going to do history from the infectious point of view. So again, in 1643, Sir Thomas Brown described an outbreak of illness that occurred in France. It was described as coarse black hairs emerging from children's skin. There was a mention of hairs or worms, and these were described as black with protruding heads. But they were also described as comedones, which are like blackheads, just a technical word for it, that protruded from the skin. The children had other symptoms too, and these were described as coughs or convulsions. Then between 1544 to 1884, historical accounts of similar illnesses describing projecting hairs occurred, and these were summarized by Kellett. So some scientists, such as Ep Mueller, thought these hairs were animate. And there's one of Ep Mueller's drawings on the lower corner over there. And it shows objects that appear to be arthropods, and including some that look like some scabies mites, and those are right around there. And other scientists, like the famous Dutch scientist Van Leeuwenhoek, he is also known as the father of microbiology, and when I was a geeky little kid, he was my hero. He said that they were inanimate. So we really have no way of knowing if these diseases were the same as what we call Morgellons today, but we see some similarities. We see some hardened comedo-like particles in Morgellons skin specimens, and then from about 1902 to 1938, there were case studies describing parasitophobias or dermatological hypochondriasis, and these were published sporadically. We also don't know if these diseases were the same as what we call Morgellons today. So interestingly, some of these early cases were linked to and involved spirochetal infection. VA reported that 6 out of 8 of his subjects in his case studies had syphilis, which is caused by the spirochete Treponema pallidum. And Ekbom, he said that he felt that there was more at play in these cases than just mental illness. And he said that finding the underlying cause of the illness was very important. Ekbom, much like his colleague VA, found that spirochetal infection was present in his patient cohort. And in fact, 3 out of 7 of Ekbom's patients had documented cases of spirochetal infection. They had syphilis. Despite that fact, and the fact that syphilis was very rare in Sweden, where his subjects were from, Ekbom did not really believe that spirochetal infection played a role or was a contributing factor. We don't know for certain if any of Ekbom's patients had what we call Morgellons today, but because he did not describe filaments coming right from the skin. He did, however, report that his patients collected samples, and he said that these self-collected specimens had little threads in them and sand-like particles. So, does that sound familiar, everybody? Okay. So, we do see similar findings in Morgellons skin specimens. So what is really important to note here is that cases similar to what we call Morgellons today were linked to spirochetal infection back as far and back as early as 1935. So do you think the CDC could have done a better job of looking for it? Okay. So in 2001, biologist Mary Leto noticed fibers in her son's skin, and of course we know she named it Morgellons disease based on the similarity to Sir Thomas Browne's. We're all getting deja vu, aren't we? So Mary sought help from the mainstream medical community, and one of the doctors she contacted, his name was Dr. Fred Heldrick, and he was Johns Hopkins pediatrician, and he was known for solving mystery cases. He examined the boy and talked to Mary, and he suggested that Mary had a mental illness. So at that point, Mary established the now defunct Morgellons Research Foundation, and she later paired up with one of our heroes, Dr. Greg Smith, and nurse practitioner Virginia Savely, and former NASA scientist Dr. William Harvey, and our wonderful Cindy Casey, and San Francisco internist Raphael Stricker. So now we get to the point. We're at 2001, and since then there's been a polarized view of Morgellons etiology, and this time we'll focus on the infection. So, in science, unfortunately or fortunately, however you might look at it, various kinds of evidence are not weighted equally in terms of value. So the strongest in terms of value are meta-analyses and systematic reviews, then randomized controlled trials, followed by cohort studies, then case control studies, cross-sectional studies, animal and in vitro trials, and finally the least important evidence are considered case reports, which you remember most of the DOP ones were case reports, and there were very few, if anything else, of the others, opinion pieces, and letters. For demonstrating a causal relationship between Morgellons and spirochetal infection, we have solid evidence in all of these categories except for maybe the meta-analyses. So what is not scientific evidence is hearsay, YouTube videos, personal anecdotes, pseudoscience websites, blogs, and patient chat groups, and websites like fakenews.com. Those are really not scientific evidence. And that includes even anecdotal evidence that comes from a physician. And it may very well be true, and it may be an important observation, and it may have value, but it's still not scientific evidence. So if a doctor tells you that Morgellons is caused by alien invasion or nanites, that does not make it so. Yes, I would recommend that. So if a provider might say that that Morgellons responds to, let's say, the treatment for delusion for an antipsychotic drug, and some patients do no benefit, they do notice benefit from that, it does not mean that it's caused by delusion. Or if they say that, well, my patients respond better to Bartonella treatment, it does not also mean that the patient has Bartonella or that the disease is caused by Bartonella. There's many reasons for that. When in true science and in scientific evidence, we need to answer a lot of questions. So, you know, how was the patient tested for this particular illness? What test was used? Was it direct detection? Was it indirect detection? How many patients were studied? How was improvement measured? So, a physician's personal beliefs might be valuable, and especially to individual patients, if they are beliefs that lack supporting data, they're still unsubstantiated opinions. And, you know, I'd like to point out that for Borrelia infection, that organism is very resistant to treatment. So, you know, there's many reasons why a patient might not respond well to Lyme treatment, even though they have it. And so when you're also treating patients with multiple antibiotics, it's very difficult to know which antibiotic and which treatment had an effect. That's why in science we do controlled experiments where we try to only test one variable at a time. So in proving that a microorganism has a causal relationship to a disease, this has been a long and difficult process. It first started the germ theory of disease, the theory that minute particles or contagion cause disease, was first proposed by Fracastoro in 1546. And at that time, most scientists believed the miasma theory, and this was the theory that bad air caused disease. Van Leeuwenhoek first saw bacteria in the 1600s, but he called them little animalcules, and he felt that they might be associated with disease, but he wasn't able to demonstrate a causal relationship at that time. Therefore, the miasma theory remained the prevailing thought of infection until the mid-1800s, and this was 300 years after Fracastoro had first proposed differently. So criteria for solving or proving causal relationships between microorganisms and disease was first proposed around 1890, and this was by Louis Pasteur and Robert Koch. Then afterwards, others like Bradford Hill and Frederickson Relman also proposed more modern recommendations for proving causal relationship. But in fact, no method is perfect. An organism might not meet all these criteria and yet may still cause disease. However, the more criteria that can be met, the more likely there is to be a causal relationship. So some people have told me, well, you know, just because you find a few bugs, that was Feldman, or just because you find Borrelia in a sample doesn't mean the person has the disease is causing it. Well, you know, that's true enough. Proving a causal relationship is a little bit more difficult, but you know, we can check off quite a few of these things, and we do have very convincing evidence to support a causal relationship between Morgellons and Borreliosis. So we're gonna just take a peek at Koch's postulates. These date back to 1890. The organism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. 2, the organism must be isolated from a diseased organism growing in pure culture. 3, the cultured microorganism should cause disease when introduced into a healthy organism. Then 4, the microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the proposed causative agent. There's a lot of problems with these postulates. So first of all, people can asymptomatically or subclinically carry a pathogen, so the disease might not occur in all those people who are infected, and disease symptoms can manifest differently in different individuals. Furthermore, not all pathogens can be cultured, and the syphilis spirochete, for example, Treponema pallidum, it can't be cultured, and it can be asymptomatically carried, and it can be really challenging to detect spirochetes in different individuals because there may be low numbers of these organisms occurring. So Treponema pallidum dismally fails all of Koch's postulates, and yet we know that it causes syphilis. So Borreliosis is consistently detected in Morgellons subjects, and Borrelial spirochetes have been cultured from Morgellons subjects and from their lesions. So we can at least fulfill two of these postulates. Now we go to the Bradford Hill criteria, and I'm sorry, but these are a little bit boring. But first is strength or size. The larger the association, the more likely it is to be causal. And so a correlation between Borrelia infection and Morgellons has been shown repeatedly. This is statistically relevant as it is not commonly found in healthy people in the general population. 2 is consistency or reproducibility. Consistent findings observed by different researchers in different institutions with different samples using different methodologies strengthens the likelihood that there is a cause and an effect. So Borrelia spirochetes are consistently found in Morgellons specimens, and this has been done in many different labs and using many different methods. Specificity. Causation is more likely if exposure causes only one disease. So this criterion is not suitable for many microorganisms. Borrelia infections are complex multi-system illnesses, and they affect individuals in different ways, and they often mimic other diseases. So secondary etiologic factors, such as co-infections that individual patients may have, can complicate the disease process. So in Morgellons, secondary etiologic factors might be immune status, age, sex, and these are likely needed for a cause and effect, and they are probably unique to different individuals. So different individuals could have different secondary factors. Temporality. The effect should occur after the disease. Borreliosis is difficult to work with this criterion, and that's because it can remain asymptomatic in patients for long periods of time, so it's not always possible to know when the infection occurred. However, we have found evidence in borreliosis in most of the symptomatic subjects we've studied. So next is biological gradient. Greater exposure should generally lead to greater incidence of effect. There may not be a simple linear gradient in some diseases. In some cases, just the mere presence of that pathogen can trigger the effect. In other infections, the pathogen may not cause disease in all people. Plausibility. A plausible mechanism between cause and effect is helpful and should make sense in light of current theories and results. There is a plausible mechanism for the cause and effect relationship between borreliosis and Morgellons. Coherence. The cause and effect should make sense with available knowledge and should correlate with clinical findings. So, for example, the histopathological evidence should support the cause and effect relationship. So we've seen in histological sections of affected Morgellons skin that Borrelia spirochetes correlate with the presence of fibers, and that's a very good example of coherence. Experiment. Strong proof of cause and effect comes from the result of experiments where significant variables are held stable to prevent them from interfering with the results. And I talked a little bit about that before, how sometimes when it's anecdotal evidence, there are no controls of different variables. Analogy. The effect of similar factors may be considered. So the researchers should accept weaker evidence of an agent causing a disease if there is stronger evidence of a similar agent causing a similar disease. Doesn't that sound a little bit like bovine digital dermatitis? So the animal disease bovine digital dermatitis is characterized by filamentous lesions, and it is caused by a spirochetal infection. As well, syphilis is a multi-system illness, and is coupled with skin lesions that bear some resemblance to Morgellons lesions, and it too is a spirochetal infection. So now we're going to go to Fredericks and Rallmans. And they published sequence-based identification of microbial pathogens, a reconsideration of Koch's postulates. And this is because new molecular techniques, everybody started working with DNA. So these revealed a lot of uncharacterized pathogens that couldn't be applied to Koch's postulates. So a new approach for showing disease causation was needed. And we have evidence that supports all Frederick and Relman's postulates, and most of this is published in peer-reviewed literature. So I still have scientists from various universities that'll review a paper I've done, and they'll say, well, your work doesn't fulfill Koch's postulates. And, you know, it's just so outdated and so sad that scientists continue to not evolve with the times. But anyway, in Frederick's and Relman's, number one, a nucleic acid sequence belonging to a putative pathogen should be present in most cases. You'll notice it says most and not all cases of an infectious disease. Microbial nucleic acids should be found preferentially in those organs or gross anatomic sites known to be diseased, and less in those organs that lack pathology. Fewer or no copies of the sequence should occur in hosts or tissues without disease. And with resolution of the disease, the copy of number of sequences should decrease or become undetectable. With clinical relapse, the copy number should increase. When sequence detection predates disease, or if the sequence copy number correlates with severity of disease or pathology, the sequence disease association is more likely to be a causal relationship. 5, the nature of the microorganism inferred by the available sequence should be consistent with the known biological characteristics of that group of organism. 6, the detection of the sequence should correlate with the detection at the cellular level. Efforts should be made to demonstrate specific in situ hybridization in areas of tissue pathology. So, you know, basically what that means is you should be able to see this stupid microorganism in those areas that are affected. And 7, sequence-based forms of evidence for microbial causation should be reproducible. So, in the case of Morgellons disease, the common denominator appears to be various species of spirochetes of the genus Borrelia. So far, 5 different labs have detected and identified Borrelia DNA from Morgellons skin and culture specimens using PCR technology followed by sequencing. And those are Australian Biologics, University of New Haven, Oklahoma State University, IGeneX Reference Laboratory, and Mount Allison University. Borrelia burgdorferi was detected by all 5, Borrelia miyamotoi by Australian Biologics, Borrelia hermsii by IGeneX, Borrelia guerinii by Australian Biologics and IGeneX. And then, using a different method called metagenomic sequencing, a 6th lab, Perkin-Elmer, detected Borrelia burgdorferi from Morgellons skin specimens. So that technique, it looks like at all of the DNA in the sample. So it finds all the pathogens. And Borrelia was one of the pathogens that showed up in all the samples that were done. The only other that showed up in the other sample in these particular group of specimens was Helicobacter pylori. So Borrelia DNA was detected in Morgellons skin specimens by a 3rd laboratory, and this was by fluorescent in situ hybridization, and this technique was performed at McLean's laboratory, and that is in New York. So experimental results from 7 labs have detected Borrelia DNA from Morgellons subjects using 3 different methodologies, and that shows reproducibility and consistency. We did a study that we published in BMC Dermatology, and this provided visual, antigenic, and genetic confirmation of spirochetes, both directly in Morgellons skin tissue and in cultures taken from a cohort of 25 patients. Different laboratory procedures were performed at different laboratories independently, and this provided corroborative evidence. So the top image shows some cultured spirochetes, and the bottom shows some spirochetes in Morgellons skin tissue. So that particular thing checks a lot of those boxes for causality. We also observe spirochetes both in culture and in skin specimens using scanning and transmission electron microscopy. And this was performed at a different lab, at Clemson University in South Carolina. We used silver nitrate staining to help us visualize spirochetal morphology, and as you can see in these images, we saw them both in tissue sections, which is at right, and in culture smears, which is left. So the spirochetes in tissue are stained black, and the tissue is gold. And this was particularly useful to see spirochetes in Morgellons skin sections. And as you can see, these Morgellons lesions contain a lot of spirochetes. It's not just that we see one or two. These lesions are loaded with spirochetes. And we performed all the staining with positive and negative controls. We used a different technique called immunostaining, and this was used to detect borrelial antigens, both in culture and in skin specimens. So immunostaining visually shows the reactivity between molecular components of a pathogen, and these are called antigens, and molecules that are produced by the immune system called antibodies. Antibodies are labeled, and this allows us to visualize that reaction. So immunostaining was performed both at the University of New Haven and at McLean's laboratories. So every experiment that we do, we use controls, and that's whether it's in PCR detection or antigen detection. So here, for example, are some of the controls that we did for showing specificity of immunostaining. This stain was reactive to Borrelia, but it was not reactive to other common microorganisms that would be encountered in skin. So the positive control is in the center, and various negative controls are surrounding on the left and right. It did not react to human normal skin with normal flora in it, or diseased skin from a psoriasis patient, or to common gram-positive and gram-negative pathogens. So when we did PCR studies, we used controls too, and controls included distilled water, skin from healthy people, normal human foreskin, and normal skin from Morgellons and Lyme patients. We also used controls inoculated from the tissues and fluids from normal healthy controls. All our negative controls were negative. We stained tissue using DNA probes that were specific for Borrelia, and this is called in situ hybridization. And this allowed us to visualize Borrelia DNA in skin sections and in culture smears. Again, these assays were performed with positive and negative controls, and this staining was performed at McLean's laboratory. Borrelial DNA was present in tissues that had Morgellons pathology. So we have found other pathogens, and others have found pathogens in Morgellons tissue by PCR technology, which detects DNA followed by sequencing. And these include another spirochete, Treponema denticola, and this was detected by Australian Biologics and by Oklahoma State University. Bartonella henselae was detected by Oklahoma State University and IGeneX Reference Laboratory. Helicobacter pylori was detected by the University of New Haven and by Oklahoma State University. and Rickettsia species by Mount Allison University. And although these organisms have all been detected in Morgellons tissue, they are not as consistently detected as Borrelia. Bartonella, for example, seems to be present in less than 10% of the specimens. The presence of other tick-borne pathogens such as Bartonella, or I should not say specimens, I should say patients. But the presence of other tick-borne pathogens such as Bartonella might be secondary etiologic factors in the development of Morgellons, or they could exacerbate the condition. They strengthen the evidence of a causal association between Morgellons and Borreliosis as they indicate exposure to tick-borne illness. Treponema denticola is a really common oral spirochete found in humans, and Helicobacter pylori is likewise commonly found in the human digestive tract. As many as 90% of the population may be infected by Helicobacter pylori. So, we're not sure about the significance of that finding. They may be secondary etiologic factors, but they also could be spread because people might pick their mouth and then it could spread to their hand and then to the open wound. So we've studied fiber origin and composition, and this has given us an insight into a plausible mechanism for the cause and then the effect. It's important to study only the specimens of filaments embedded in skin as seen here, because I found that when I study unattached fibers, they often turn out to be environmental contaminants. Wounds are sticky, and environmental fluff and debris can stick to it, and sometimes patients think it's come out of the skin, but when I actually analyze those specimens, it's often environmental junk. So, at the base of attachment, fibers are nucleated and they're continuous with the surrounding skin cells. This shows us that they have come from those skin cells, and at the base of attachment, it shows that they're actually composed of these skin cells. And then as the fiber grows and older cells are pushed upward, they lose their cellular content. So you can see the base of attachment there and how there's the little blue nuclei, and you can see that this fiber is continuous with the skin. This fiber actually has a little branch in it too. So we know exactly what cells these things come from. We also did some special staining called Gomori trichrome, and this showed that these filaments are composed of collagen and keratin. So keratin is always stained red and collagen is stained green, and collagen is produced by fibroblasts in the dermis, and keratin is produced by keratinocytes that are in the basal cell layer. We also know a little bit about the blue coloration, why fibers are blue. This turned out to be because the blue is due to melanin pigmentation, and melanin is the pigment that humans produce that produces sort of black or red coloration. Pheomelanin produces red coloring, and eumelanin produces dark coloring. So we took some of these blue fibers and we did a stain called Fontana Mass, and that turns melanin black, and sure enough the blue fibers turn black. So they are melanin. And the red ones didn't react, but I really don't know. Maybe pheomelanin reacts different. I'm going to have to check on that. And at present, the red coloration remains a mystery. So like I said before, reproducibility is really important in science. So I sent a very famous scientist some fibers, some blue fibers for analyses, and I asked him to determine what was causing the coloration and to study them. And this scientist was at the University of Akron. I didn't know him. I found him as an expert on colored fibers and things like that by Wikipedia. So he didn't know me and had no reason to report in favor of what I wanted him to. But he reported that the blue pigment was melanin pigmentation too. And he did this because he did electron microscopy and he could actually see the melanocytes and he could see melanin granulation. And he also did spectroscopy, and this also verified the findings of melanin and keratin. So he said that the particular samples I sent him had hair-like morphology, so the scaling on the cuticle of these fibers was very much like it was consistent with human hair. So those particular blue fibers I sent him were actually very small hairs. And so he came up with very similar conclusions to ours using different methodology. So there's evidence of defective keratinization in Morgellons lesions, and I talked a little bit about these comedo-like things, and we heard that early scientists also described these comedo-like or sand-like granules, and these can form within pores. They're composed of keratin, and they feel a little bit like sand, and unmagnified they look like sand, but when you look at them under the microscope, they're consistent in shape with a comedo. So, and they can have fibers attached to them or protruding from them. And if these form when it's been inside a pore, the filaments can grow around and around because they're within the confines of that space. And you can see that on the bottom right over there. And on the middle one over here, there's some colored ones. So some of these samples, some patients produce hyperkeratotic follicular casts, and this is where the follicle or the pore thickens and becomes highly keratinized, and the keratin layer just thickens and grows and grows. This is a really common finding, and it indicates defective keratinization. These two can also have unusual filamentous attachments, and there's some arrows pointing to those. Again, if this forms within a confined space of a hair follicle or a pore, it can wind around and cause a little wad. When we sectioned some of these, we found that these keratinized projections or follicular casts are composed of keratin and collagen, and you can see that by the Gamori staining there. So they're red and green, keratin and collagen. This is a particularly interesting photo. You can see a fiber cross-section, and then right beside it, some immunostained Borrelial spirochetes. So many times we see the actual filament in close association with the presence of Borrelial spirochetes. The strong association between Morgellons and Borreliosis has been shown in several studies, and again using different methods. Savely and Stricker reported that 96.8% of their cohort of 122 subjects had a Lyme disease diagnosis. Middelveen et al., that was the BMC study, 2015, showed that a cohort of 24 out of 25 Morgellons subjects had detectable Borrelia DNA in Morgellons specimens, and that all 25 had evidence of spirochetal infection. Fessler 2017 showed that in a cohort of 60 Morgellons subjects, all of them had serologic laboratory evidence of Lyme disease. So two independent studies of Lyme patients, one by Main et al. 2011 and one by Fessler 2017, showed that approximately 6% of Lyme patients have Morgellons. So you might ask, well, why don't all Lyme patients have Morgellons? Well, why don't all Lyme patients have arthritis? Why don't all Lyme patients have seizures? Why don't all Lyme patients have ACA or lymphocytoma? You know, different patients have different symptoms. And that's because there's other secondary factors or risk factors that need to be present for these symptoms to occur. And these might include things like the strain of Borrelia, genetic predisposition of the patient, the immune status of the patient, hormonal status, age, general health, presence of co-infections, and maybe even some factors such as skin damage. I saw fibers form on one patient who'd been out in the sun and had a sunburn, and that's where she got her fibers. And personally, I think that's maybe why some of the southern states have a higher incident rate, because there's higher incidence of sun exposure. It's possible, too, that other spirochetal pathogens, such as treponemes, may be the principal causative agent in a subset of Morgellons subjects. I have not observed this yet. We have detected Treponema denticola, but when we did, it was in the presence of, it was co-infecting with Borrelia burgdorferi. But theoretically, this is possible, and I suppose it is possible that other unknown pathogens could produce a similar effect. We don't know everything. And it's always important to be humble and realize that you don't know everything. There's more evidence of causality if the focus of known infection is the same as the focus of the effect. So in the case of Morgellons, we don't know much about the incidence rate or the geographical distribution. So the now defunct Morgellons Research Foundation reported in 2012 that most of the self-reported cases occurred in Florida, California, and Texas. And I've sort of colored these in, actually Dave did it for me, thank you Dave, in dark blue. And the OSU registry that Dr. Randy Wymore had, he reported that most of the cases were occurring in the states of Oregon, Nevada, Arizona, and Oklahoma. So these distributions don't really match the acknowledged distribution of human Lyme disease cases, and those are mostly in the New England states and around the Great Lakes, as seen over here. There's a lot of different reasons for that could be responsible for that disparity. And first, the geographic area of infection might differ from the area where symptoms develop. So as symptoms might not develop until long after infection occurred. For example, a person infected in New England might later retire to a southern state and where they develop Morgellons after a long period of being asymptomatic. Secondly, multiple Borrelia species are implicated in Morgellons, and their distribution differs from that of Lyme disease. And we can see that in this map of Borrelia hermsii, which is up there. Another reason, there could be bias in voluntary reporting. So some states such as Texas, where this conference is held, may have greater awareness of the disease. So therefore, self-reporting may be more frequent in Texas. And finally, I'd like to point out that our knowledge and our maps may lack accuracy, both for Lyme disease and Morgellons disease. And I'd like to point out that canine Lyme disease maps differ significantly from human Lyme disease maps, which I will show you. So this is the map for canine Lyme diseases, and you can see that there's quite a few cases in Texas, California, Oregon, Oklahoma, and Florida, as well as in the New England and the Great Lakes area. So that's quite different than what we saw for human cases. And it's a little more similar to what we see for Morgellons. But then, of course, we know that Texas doesn't have any of this, but the dogs didn't read that. So lastly, remember I said that, weaker evidence should, if it's supported by stronger evidence of a similar condition, has better evidence for causality and should be considered. So we know that a Borrelial filamentous skin condition was reported in a canine model, and dogs in this study had red and white and blue filaments, and these were composed of collagen. And they also had defects in keratinization, such as deformed hairs and these thickened follicular casts that we see in the humans. They too had detectable Borrelia associated with their diseased tissue. So an analogous model provides evidence of a causal relationship. So again, here's another analogous animal model. So there's similarities between Morgellons, which is seen in humans, with the animal model bovine digital dermatitis, which is seen in cattle. This fact strengthens the evidence of a spirochetal relationship for Morgellons. In the cattle, the cattle have these lesions above the heel bulb, and those lesions have unusual keratin projections, and the tissue is infected with spirochetes called treponemes, and you can see the infected tissue with all the little black squiggly spirochetes in it. And they're abundant in the diseased tissue, much in the same way we see abundant Borrelial infection in Morgellons. So experimental infection of calves was performed, and this was done with pure cultured treponemes, and it caused disease in the animals in an experimental infection and showed a definitive causal relationship. So that analogous animal model even satisfies old-fashioned, outdated Koch's postulates. They can get it on their udders, and the treponemal infection is systemic, but cattle stand a lot in the dirty conditions seem to aggravate the skin. They probably get micro, where the skin gets damaged and they're in a really wet environment is where the lesions tend to develop, and you can find just like the diseased tissue is full of spirochetes. So, but you can also isolate the same treponemes from manure and from other samples in these. So they too have a systemic disease. So as far as mechanism is concerned, it's a well-established fact that infections can cause abnormal keratin expression and keratin proliferation. Epstein-Barr virus causes a condition called hairy leukoplakia, and this occurs in HIV patients. So this white hair-like keratin growth occurs in the mouth, mostly along the tongue, and you can see it up there. So we know pathogens are capable of doing similar things. Cholesteatoma is a condition in which filamentous keratin growths occur in the ear, and this is caused by an inflammatory process due to infection from various different bacteria. So, we see infections can cause unusual filaments, so I don't know why the hell everyone's so surprised that you guys might have an infection. Makes no sense to me. It's really ridiculous, isn't it? Mind-boggling. So, the study by Aberer et al. Showed that Borreliosis enhances collagen expression, and it can stimulate growth factors responsible for increased collagen production. Our study showed that most of the Morgellons fibers are actually composed of collagen rather than keratin. I mean, we do see both, but more often I see these collagen fibers. And you can see it in this image down here. So all those little squiggly things are sections of Morgellons fibers, and they stain green in Gomori, so they're collagen. Finally, we know that Borrelia can invade both keratinocytes, which are the sites that produce keratin, and fibroblasts, which are the cells that produce collagen. And so we know that they're capable of infecting those cells. And lastly, I want to end by comparing our evidence with the evidence that was presented in the original study, concluding that there was a causal relationship between spirochetal infection and Lyme disease. So back in the early 1970s, a multi-system illness was associated with a bullseye skin rash, and this was reported in Connecticut. There was geographic clustering, and the setting was rural, and this suggested that maybe an arthropod vector might be involved. So this study by Steer et al. In 1983 reported achieving 3 positive spirochetal cultures from skin, blood, and cerebrospinal fluid from 3 subjects. But this was out of a total of 142 samples taken from 56 subjects. So they were far less successful at doing this than we have been. 2 positive cultures were achieved from samples taken from 110 Ixodes scapularis ticks, which was at that time called a different name. So they also had some serologic evidence. So they had 40 patients that had EM rashes, and 90% of these patients were IgM positive, had reactivity. And they had about 95 patients that had later illness, and they reported that 94% of these were IgG positive. The cultures taken in this study were characterized only by immunostaining, which was a method I described, and there was absolutely no DNA evidence. And this was quite a long time ago, where it was much harder to do DNA work. This study concluded that the spirochete they isolated was the etiologic agent for Lyme disease. So I'd like to point out that at this point, we have a lot stronger evidence for a causal relationship for Morgellons and Borreliosis. Thank you so much for listening, and I hope that last one wasn't quite as entertaining as the delusional one, so I thank you for your attention. And thanks to Cindy Casey and the Charles E. Holman Foundation. Thanks to all my co-authors and my co-investigators. Thanks to Virginia Savely, Peter Main, and Randy Weinmore for all their pioneering work. And to IGenics, and also Drs. Vet Lloyd and Julie Lewis for their work on Morgellons and for collaborating with me. And most of all, thanks to all of you Morgellons patients. And thanks for all the generosity in giving me samples and also sharing your knowledge of this disease. Oh, and one more boring test to perform. You have to know that I have my references, so you know that I'm a real scientist. Otherwise, you might think I really don't do anything. Okay, there we go.